Hello and welcome to today's webinar. On behalf of Drug Target Review and Biosensing Instrument, I'd like to thank you all for attending. I'm your moderator, Nikki Withers, and I'm the editor of Drug Target Review. Today's speakers will be Dr. Nicholas Larson, who is the Associate Principal Scientist at AstraZeneca, and Dr. Shiji Wu, who is Application Scientist at Biosensing Instrument. Following their presentation, we will move on to a live questions and answer session where you can pose questions to today's speakers. Please remember you can submit questions at any point during the webinar using the questions panel situated in the menu on the right. Before we begin the presentations, I'd like to invite you to participate in the following polls, which should be launched on your screen. So the first one, what is your application? So we've got four options: small molecule, biologics, both, or other. You can just click which one applies to you. Perfect. And then we do have another poll as well, which should be coming up shortly. So our second one, which tool do you use for affinity or kinetic measurement? And there are three options on this one for you. And if you could just click which one applies to you. Perfect, thank you so much. So without hesitation, may I pass over to today's keynote speakers, which hopefully you should be able to see now, Dr. Shiji Wu and Dr. Nicholas Larson. Thank you.
Okay, first of all, thanks for hosting, and thanks for everyone to join us. So I guess I will share with everyone today the basic principles of this new technique, the surface plasmon resonance microscopy, and some of its unique applications. So before I start talking about the SPRM, so let me say a couple words about a biosensing instrument. So we are the bioinstrument manufacturing company, and we are located in Tempe, Arizona since 2005. And we have developed a series of SPR instruments and with unique functions, aiming at the applications in bio research and also material science, and particularly in 2017, and we actually introduced this new technology, the SPRM, and we have received a wide interest from bio research institutes all over the world. So the SPRM is actually designed for study the binding properties of drug molecules with membrane proteins in cell. So you may wonder, why do we study this membrane proteins? And one of the big reasons membrane proteins play such an important role in the drug discovery, and it is estimated that over 60% of the drug targets today are membrane proteins. And on the other hand, the SPR is a very well established, effective way of measuring binding kinetics between different molecules. It's kind of natural for one to trying to use the SPR to study the binding properties between the drug molecule and membrane proteins. However, this membrane proteins is not easily be this experiment cannot be easily done, and partly because the conventional SPR requires these proteins to be purified and fixed onto a sensor chip surface with uniform. And the membrane proteins, on the other hand, are kind of fragile and they cannot be easily purified, and they kind of tend to lose their activity significantly during the process because of losing their native environment. So the challenge here is how to be able to use SPR to do the kinetic measurements while keeping the native environments for those proteins as closely as possible. So SPRM is kind of designed just for this purpose. So what is the surface plasmon resonance microscopy? That's SPRM. So SPRM actually has 3 key components. So the first one is the SPR components, or includes the light source, the detector, and also the sensor chips. And those are for the SPR measurement. On top of this SPR components, and we have a bright field optical microscope. And this one is used for identifying the sample and positioning the sample for the measurement, and also to monitor the stages or the state of those samples during the experiment. And the last component, which is also very important, critical components, are a specially designed microfluidics system. And this microfluidics system allows the proper flow conditions for the SPR measurement, and also maintains the physiological conditions for the cells and the membrane proteins. So how do we actually do the measurement on cells with surface plasmon resonance? Well, unlike the conventional SPR, we actually, in SPRM, we use a collimated light source, which is actually illuminate a fairly large area of the chip surface. When the each spot on the chip surface will have a unique SPR reflection angle, which is also the SPR signal, the response signal. And when the surface is uniform, and you will get one signal for the same signal for all over the surface, the illuminated area. And however, when the surface becomes heterogeneous, which is non-uniform, such as you have a cell on the surface, and you start to see variations of the SPR responses at different spots on the surface and due to different surface properties. And when you map these variations of the SPR signal as a function of the lateral position, and you will get an SPR image that corresponds to any particular time. Now, say when you are introducing the chemicals into the solution and have cells on the surface, and when those chemicals start binding to the proteins on the cell surface, and this will change the surface properties and also for the SPR response. And in result, and you will see a change in the recorded SPR image corresponding to the binding process. And this whole process of binding between the chemical molecules and the surface proteins can be recorded in a series of images as a function of time. And this whole process can be recorded, and after you finish the experiment, you can go back to the images and pick any of the positions on the spots on the image and extract the recorded SPR signal and backplot them or process them to plot them as a function of time, and then you will obtain this so-called SPR sensorgram, which corresponds to the binding and the association and the dissociation of those chemicals and the surface proteins. And by analyzing this sensorgram, we call the sensorgram or this spectrum, and we can obtain those kinetic information such as the K-on for the association and the K-off for the dissociation and the affinity of the equilibrium of the binding. So this is for actually in principle you can you have an image and you can actually get a data point for each pixel on the image. But in practice, the analysis is done in a much more efficient way. We call it the objective data analysis. So in this process, we actually divided the whole area into a certain number of regions of interest. We call the ROI, the region of interest. So each of the small squares, and the squares, the number of the squares, the small regions can be controlled by the customer, put into the system by the customer. And each of the small regions acts kind of like a channel on a conventional SPR. So each of the ROI, or the region of interest, will give you one set of sensorgrams for all the concentrations you did the measurements. And then we will apply the first order binding models to the sensorgrams and to do the fitting and do the analysis. And all those ROIs, all those sensorgrams that doesn't really fit well with the kinetic model, which is corresponding to like non-specific bindings and also like some other secondary effects or whatever, and they will be zeroed away. So whatever left on the surface are those good sensorgrams and fits well with the binding kinetic models when corresponding to specific bindings between the molecules and the proteins, and will be left over, and then eventually we'll have statistics over all the data obtained, and from that we'll get the average value of the binding parameters like the affinity and the K-on, K-off, and also because of the ROIs covers the whole area, and the surface is not uniform, and we see the heterogeneities of different parts of the cells, and that's why we see the distribution, and which reflects the cell heterogeneity and the receptors on the cells. Now, this is basically the principles of how the SPRMM works and how it does the measurement and how do we analyze the data. So this example here will show you is one of the early experiments and a kind of typical experiment to show the workflow, how this experiment is done. And this system is actually looking at WGA, glycoproteins and sugar binding sugar residues on the cell surface. And what is unique here is if you look at it, this is actually working with a single cell. So the whole area is roughly about like 80 microns. And so I have a video here to show you. You can look at it. So for the experiment at the beginning, so you have a pure so you have a pure liquid, so you have a baseline, and then you introduce the WGA, you start binding, you see the signal changes, and then you switch off the injection, and you start to dissociate, And the last jump in the signal is caused by the a standard process called the regeneration. And after that, and all the sugars was removed from the cell, and then the signal basically go back to the original baseline. So you can see the whole process is recorded in this whole series of images, and the image shown here is actually taken off the pile, and which is recorded as 550 seconds during the process. Is somewhere around here. It's almost at the end of the dissociation. And just as we said, with all those informations, we can generate all those sensorgrams and do the analysis and to obtain the kinetic parameters. And for example, here we actually can get a two-dimensional mapping of the like the K on and the K off along the different regions of the cell. Now, so most of the applications for SPRM is actually with small molecules or antibodies. So I will show a couple examples with each one of them. The first example I want to show you is a small molecule targeting an NTS1 receptor. And the three images shows here is the one in the middle is actually the bright field, the optical image of the cell surface studied, and then the right side is the SPR image recorded during the experiment. And those ROIs, the color squares on the image, are those the regions which is active regions after we did the filtering with the kinetic model, and which will give the our final results for the parameters like the affinity and the K on and the K off. And the first image on the left is actually an ICC fluorescence image to validate the binding between the small molecule and the receptor. So the affinity will we measured using SPRM is reported about 2.03 nanomolars in this case. And this is, sample is actually being studied very well by a number of researchers. For example, Dr. Harper from Roche have purified this protein, this NTS1 receptor, and studied this binding with conventional SPR. The results for the affinity reported by Dr. Harper is around like 400 picomolar. And there's also in the literature some in vivo experiments which gave some gave the affinity as around like 8 nanomolars. So you can see the results from the three different measurements. They vary a little bit, but in a sense, they all make sense. So this second example with small molecules I want to show is the we have done with Genentech. And this is, again, a small molecule. And we have, with the same process, we have seen a very good distribution. And the average affinity obtained from the experiment is 865 nanomolars. And in this experiment, we have actually used both the negative compound and also a negative chip as negative controls for the experiment to ensure the when we are looking at the real bindings between the molecule and the protein. So this is another example. It's done with Daiichi Sankyo in Japan. And this is histamine binding to GPCR on a HEK cell. And on the left, we have the optical image, of course, and the SPR image. And this SPR images actually show the the active binding sites, and it actually corresponds very well with the cells on the optical image. And on the right side, those images are done with the mutant histamine, which used as a negative control. And with the negative control, we can see much lower or very little, or almost a very low level of binding signals, which is confirms the validity of the experiment. So the numbers we measured get with this experiment for the KD affinity is about 7.4 nanomolars. And again, this is small molecule histamine, and the molecular weight is only about 500 Dalton. This experiment is interesting. It's actually designed to measure the to try to find the mechanism, for drug resistance. And the system used here is Herceptin and HER2 binding in human cancer cell. And the fluorescence image here shows the HER2 receptors are overexpressed on the cell. And this is one of the SPR image recorded during the experiment. So the top sensorgram here is generated from the areas on the cell, and this is one in the empty area without the cell. And you can see there's many of the sensorgrams corresponding to the cell, and then we have one average sensorgram from all those individual ones, and then the red curve is actually a fitting for the averaged sensorgram, and from which the kinetic parameters is measured. The bottom sensorgrams are the sensorgrams on the cell generated with different concentrations of Herceptin in the solution. And this experiment actually goes on to be able to help to identify the third-party third molecule blocking actually being the major reason for the drug resistance in the system. Now, so it's the most of the experiments we've been done can be done with the cell lightly fixed in the system. However, many people working with antibodies are kind of reluctant to fix the cell because of they're afraid of changing the activities of those receptors to the antibodies. And so the question is always, can we work with live cells, without further doing anything like fixing it or not? So this experiment we done with one of our partners in Europe, and the it's actually try the experiment with active cell in medium. That's a CO2 independent medium. And if you look at it carefully, the in the buffer bottle, this fluid is actually reddish. Okay, so all I want to show in with this example is this to confirm with you yes, it's possible to work with live cells and directly working with the mediates with this SPRM system. And of course, with this experiment, we can still obtain the kinetic parameters. For example, the affinity for this system we have obtained is about 1 nanomolar, and also the K on and K off. And another question people always ask is, it's easier to work with adherence cells, but what about those live suspension cells, and which doesn't want to stick to the surface and tend to floating in the liquid? And yes, we have actually finally we've been worked with one of our partners in Pfizer, and we have developed this protocol and be able to grab those live suspension cells from the liquid, from the solution, and hold them onto the surface and proceed to do the SPR measurement, the binding measurement. And this example shows here is actually done with WGA. It's not antibody, but this is used as a control experiment or test experiment. So what we see here is that we've been able to successfully to hold the suspension cells onto the surface and then do the measurement. And of course, the measurement for the WGA here is affinity is about 7.5 nanomolars. And what Pfizer interested is actually continue the work with this protocol and to go do antibody binding measurement. Okay, those are the applications with cells. Actually, SPRM is actually a unique technique. It can do something other than working with cells. It's actually some other interesting application. Here I want to show you one of them, just for curiosity. And we know the surface plasma is like electromagnetic waves or energy resonators traveling on the surface of the sensor chip. And when the surface is free, it travels freely. And when you have a particle, a nanoparticle on the surface, and then this travel of those plasma waves will be blocked and interfered, and you will start to seeing scattered patterns of the plasma wave caused by the nanoparticles sitting on the surface. And as a result, you will see this very unique patterns, like a boat, traveling in the running in a calm water. And those patterns, the size is much bigger, than the comparing to the nanoparticle itself. With this, we cannot see the nanoparticle, but with this pattern, we can actually identify the nanoparticles on the surface. And you can actually calibrate the system to measure the size of the nanoparticle and also the distance of the nanoparticles away from the surface. So one of the applications for this is to look at the effect of antibodies on a bacterial cell. So the cell here is E. coli, and it sits on the surface. It's actually tethered and by some linker. And when the bacterial cell is alive, and it actually jumps up and down along the surface, along the Z direction. And the result is actually the you will see the pattern of the SPR image. The intensity of it changes periodically. And when you introduce the antibodies into the system, and the bacterial cell will become quiet and starts sitting on top of the chip surface without moving anymore. And then you will see the intensity of the pattern become constant. So the picture just shows you now it's alive, and now it's actually after the antibody is introduced, so it's just sitting on the surface. So with this experiment, you can actually study the how those effects of those antibodies on the life of the bacterial cells. And of course, this the numerical data and the statistics from all the measurements. So in conclusion, and SPRM is a unique technology, allows you to work with membrane proteins, in its native state, and it's a label-free technique, and for kinetic binding measurements, and you can not only give you the kinetic measurement, but also can provide you the lateral information about the differences between cells and the different part of cells, and work with cell heterogeneity. So with this, I will pass the time to Dr. Lassen for his amazing work on membrane receptors. So Dr. Lassen, is all yours.
Hi there, Nicholas. I think you just need to put your sound on, please.
I'm really sorry about that. Let's see here if it works now.
Yeah, I can hear you. I can hear you now, Nicholas.
Okay, I'm really sorry about that. I make a restart here, and I'm really happy to be here and talk about a case study that we have performed on GPR39, which we have used for a pilot evaluation of label-free binding assays here for membrane receptors within AstraZeneca. Okay, a little bit of introduction to GPR39. GPR39, it's a family A G protein coupled receptor that belongs to the ghrelin subfamily, and all the subfamily members here, except GPR39, they are established peptide receptors involved in regulation of metabolism. And it was a reasonable hypothesis that GPR39 would be similar, and actually quite early on, the peptide orbistatin was reported as a ligand here. But that finding was never confirmed, and it was actually later it has only been shown that the metal ion zinc is the only endogenous agonist for this receptor here. GPR39 is quite widely expressed, but mainly it's found at the highest levels in endocrine metabolic tissues, and it has been implicated in several different physiological functions. But I should say that the physiological role is still being elucidated, and this progress into this has been quite slow due to the limited availability of pharmacological tools here. I think among the most convincing evidence now is that GPR39 agonism is resulting in somatostatin release, which in turn inhibits the hunger hormone, ghrelin. So you can clearly see that there is a link between link to regulation of metabolism here for this receptor. GPR39 signals via multiple pathways, and I will show you that on this slide here. So first, talking about general GPCR signaling here. So downstream of GPCRs, there are 4 major different classes of G proteins that are transmitting signals via completely different types of signaling pathways here. In the case of GPR39, it is actually able to signal via 3 of these different pathways. And in the study that we have performed, we have measured functionality of the GS pathway by measuring cyclic AMP. Via the GQ pathway, we have measured inositol phosphate metabolites. GPR39 also signals via G1213 here. We haven't directly measured the effects of G1213 here, but what we have done is that we have measured the integrated cell response, and we have done so by measuring dynamic mass redistribution here. And I won't go into details of that, but essentially what it is that we have been using the Corning EPIC instrument that is sensing changes in refractive index as a consequence of cytoskeleton and other intracellular rearrangements. So we can measure essentially responses via 3 different types of pathways. I should say here that we also looked into opportunities, of course, to look for binding here. And we didn't have radioligand binding easily accessible, so we tested first backscattering interferometry as one technology to address this here, and later on, of course, also the SPR microscopy here. To identify compounds, to identify agonists for GPR39, we performed an HGS by measuring in a functional assay. We measured cyclic AMP responses. And then I'm pointing here on the right-hand side on the AZ1395 compound. And you can see the green curves, that's representing concentration responses in the cyclic AMP assays. And here are 3 different compounds or chemotypes, example of compounds from different chemotypes here that you identified in the screen here. Focusing just on 1395 here, you can see that we have a potency here around a little bit better than a micromolar in the cyclic AMP assay. And this is done on HEC cells, HEC293 cells overexpressing the GPR39 receptor. When we are looking at another pathway, exemplified by the blue curve here, looking at inositol phosphates in the same cell, we see quite a large potency shift. So clearly the compounds are much more potent when looking at signaling via the inositol phosphate calcium release pathway. And also to further illustrate the complexity when it comes to GPCR pharmacology and functional assays, looking at the black curves here, that is the same type of assay, the inositol phosphate assay, but in another cell type, in NIT1 cells that are endogenously expressing GPR39, but at much lower levels. And then again, the potency is much, much lower than compared to the overexpressing cells. So I guess my point here is that agonist potency as well as efficacy depend very much on the cell system and the pathway component that you're measuring in, and does not really inform on affinity. So let's see if the next slide comes up here. Yeah, so what we also saw, since zinc is an agonist for GPR39, we also looked for allosteric modulation, and we saw that the compounds are clearly very much modulated by zinc. So in the absence of zinc in the dynamic mass redistribution assay, we have a potency of approximately 3 micromolar. That is shifted approximately 1,000-fold in the presence of high concentrations of zinc down to a quite impressive 3 nanomolar potency. So I think that from the functional assays here, we have quite a decent understanding of the behavior of the different agonists here, but we of course want to know how this relates to the binding affinity of the compounds here. And here, when we performed these studies here, we really didn't have access to a radioligand here, and I think this so we couldn't easily perform those assays, since radioligand binding, that is really the general way that these type of studies are done here on membrane receptors here. So this is to illustrate that it's not really straightforward when radioligand binding is not an option. So I don't need to mention in this audience that SPR is very challenging here on membrane receptors. Another option is single molecule microscopy, but that has another drawback, that it requires development of ligands that can retain affinity after fluoro labeling. There are emerging technologies like cellular thermal shift assays, where examples are starting to come from membrane receptors, but it's still a technology very much in development. So in this case here, we tried backscattering interferometry, which has been shown to work at least on membrane preparations. And of course, we tested now more recently SPR microscopy here together with biosensing instrument. So some time ago, we measured affinities using backscattering interferometry together with a small company called Molecular Sensing, and measurements were performed in their labs. And this small company doesn't really exist anymore here. Nevertheless, we were able to obtain decent binding data here, and as you can see on the example curves here, we have affinities for these 3 compounds in a range from 50 to 250 nanomolar. What I would like to point out is that we also tried measurements using zinc alone without the presence of compounds. And this was done in a quite large number of replicates here to make sure that we are not seeing just artifacts here. But the binding, the data really suggests that we have 2 different binding sites for zinc on this receptor here. One higher affinity site with approximately 1 or below 1 micromolar, and one low affinity site here with a few hundred micromolar affinity. So after the small study with backscattering interferometry, we have continued to look into options here or opportunities to perform label-free binding measurements for membrane receptors. And obviously, we got in touch here with biosensing instrument here, and we performed a small pilot study together with them here on the same molecules here. And the cell work was performed in the AstraZeneca labs, and the SPR measurements were performed by biosensing instrument. So here is say the first example here. So we used our HEC293 cells overexpressing GPR39, cultured them on collagen-coated chips, and fixed them with paraformaldehyde directly on the chips. Then we shipped the samples over from Sweden to US. Chips and compounds were stuck in customs for a few weeks. Then they were stored in fridge at the biosensing instruments for a few weeks before the actual SPRM measurements were performed. As you can see, it's quite a small compound, molecular weight of 333 here. And yes, she has nicely gone through the analysis of how the analysis is done here. So just here for you to show you how the SPR and bright field images of these cells are looking here. Okay, so here on the top left on the slides, here are the concentrations from where the compound has been run in several different concentrations here, and you see the KD analysis for all the different regions of interest, and you have the corresponding KD histogram for all these regions, and we land on an affinity that is approximately 400 nanomolar. And when we are comparing that with the affinity that we get with backscattering interferometry, it's say 413 instead of 233 in this case here. So right or wrong, it's at least it's values that are in the same ballpark using these two different methods. Here are similar, or here is the corresponding analysis for the other two compounds, where we land on affinities for 25 nanomolar for the 4237 compounds and approximately 6 nanomolar for the 7914 compound. Of course, we want to make sure that this binding is really due to GPR39 interaction, and we tested that by using untransfected 293 cells that are lacking this receptor here, and we could barely detect any SPR signal at all there. So it looks really that it's GPR39 specific. We also performed a kinetic analysis here, and you can see some example sensograms here on the left-hand side from some of the regions of interest out of approximately 450 regions. And also then on the right-hand side, the association rate histogram here for all the regions, as well as the dissociation rate histograms here. And when deriving the KD from these kinetic data, we obtain a KD value of 617 nanomolar to compare with the equilibrium measurements, which were landing on 413 nanomolar. So just a little bit of summary here. I won't go through all the different numbers here, but from equilibrium measurements here, when we are comparing the KDs obtained with SPR microscopy with those from backscattering interferometry, for two of the compounds, we obtain potencies that are, say, within twofold from each other. For the third compound, the values clearly don't match each other, and also we had problems to obtain data, kinetic data here for this third compound, so non-conclusive data there. So something fishy is going on with that compound. Maybe it has some kind of other mechanism of action or slightly different binding site or so. We simply don't understand what that depends on here. And again, referring back to, say, the functional data, I want to stress that it's really important to obtain the affinity measures here, since the potency data is very much depending on the cell system, and it can also depend on, say, coupling efficiency towards different pathways, and that different ligands can have different bias towards different pathways. So I feel much more confident about data, comfortable about taking conclusions for different chemotypes if I can complement the functional data with binding affinity data. So as a summary here of this, I won't read through the whole slide here, but I think the key message is that we managed to obtain label-free small molecule binding data here using fixed whole cells. And I realized that this is a very small study that we have done with one receptor, 3 compounds representing 3 different chemotypes here. And I would really very much like to see larger studies where we have many compounds from the same chemotype, also comparing data from using different methods, using, for example, comparing radioligand binding, so that we really can see correlation between SPR microscopy and that type of method. Yeah, and I'm really looking forward for an instrument here, where we have easy access to a technology here for label-free measurement of small molecule binding to membrane targets, so we don't need to use radioligand binding, or when radioligand binding is not accessible. And with that, I would like to thank the people, both from AstraZeneca and from our collaborators here that have contributed. And in particular, I would like to thank Maria Flocco, that has been instrumental for setting up this collaboration. And of course, I'm very much grateful for all the help that Win, Shiji, and Akimi has provided from biosensing instrument. Okay, thank you very much.
Thank you, Nicholas. So thanks very much. Those presentations from Dr. Nicholas Larson and Dr. Shiji Wu. So thank you very much. They were excellent.